All right, y'all, let's get started. Okay, a couple of announcements for today. So I'm still going to announce this every time we're in class. Exam 1's next week, Monday. If you're ready, my office hours are today and tomorrow. And again, if you are interested in me scheduling office hours for another hour later on in the week, send me an email early in the week, please. The makeup policy for the exam is basically tell me as soon as possible, and we will try to get you scheduled for an exam either beforehand or right afterwards. But within 2 days, hopefully, we'll have everyone take that exam. And the reason it has to be so strict in that is because I hand your exams back to you, and I tell you what you did right and what you did wrong, so you can learn something for the final exam. And I don't think that actually happens in all the different classes here. Darwin Day is tomorrow, and they're still looking for volunteers if you're interested. So send me an email, and I'll connect you if you want to go pass out random knowledge on evolution on the street tomorrow. And Academic Mentoring Matters, James came in on Friday. And remember, there's always that resource with a bunch of people that have gotten A's in this course before, over on the far side of the building over there in that classroom. Anything else needs to do? I think that's it for today. So I'll give you kind of a plan going forward for the next couple days before the exam and what actually will be included on the exam. Today we'll be finishing up Chapter 5 for gene linkage and recombination. On Wednesday, we will be doing some QTL mapping exercises, which is not in your book whatsoever. This will be something that's only really available in lecture for talking. And on Friday, we'll be covering Chapter 6. Chapter 6 is talking about polyploidy and what happens with chromosomal mutations as well. And it'll be in a very limited general variety on the exam on Monday. I'm not going to put very much on the exam from that. And if it is, it'll be fairly general. Okay, so let's start with gene linkage recombination. Any questions about schedule or what's going to be on the exam before I start? Excellent.
Oh wait, I have a question.
Yep.
Like you said the other day, okay, so are there going to be some questions that we don't answer?
On the exam, it'll be completely clear when I have a multiple choice question that select all the answers available, and when there will be ones that are just one answer where it doesn't penalize you to guess whatsoever. And I'll make that incredibly clear on the exam. And the quizzes from now on as well, too. Okay, so let's pick up where we left off on Friday. We're talking about gene linkage and recombination, crossing over, and the different impacts it's going to have on genetic variation in multiple generations here. So if we're talking about two genes, A and B, that are on the same chromosome, and there's no recombination whatsoever in meiosis and in prophase 1, then we go through here, the gametes that we can actually create in that generation are going to be big A, big B, and little a, little b, little a, little b. These are all non-recombining gametes right there. We have to have some sort of recombination to be able to get chromatin with big A, little b in here, to be able to get those other genotypes out of this. Now remember, this is typically what we think about in an F1 generation right here, because you have these two chromosomes that came from the parental generation up here already. You have already those two homologous dominant and homologous recessive chromosomes coming from mom and dad there too. So you got a picture that we're taking these gametes from one individual, but we're either selfing this individual or we're crossing it with another individual in a dihybrid cross that has the exact same phenotype. So if we cross this individual with another individual with the exact same phenotype right there, also with no recombination as well, we can never get a gamete that's big A, little b, or little a, big B without recombination. These are on the same chromosome, they are linked. The key point of this going through is that when we do have recombination, we can come and get some of those recombinant gametes that we have down there, where we get the big A, little b, and the little a, big B coming through here. And remember, with a single crossing over event, this only occurs in 50% of the gametes. So the recombination rate is less than the rate of crossing over here. Now remember, between any two genes on a chromosome, recombination doesn't happen that frequently necessarily. So if we're looking at the next generation, we cross this individual with either double recessive homozygote in a test cross, or an individual that doesn't have any recombination together, what we get out of it is far less of the different recombinant genotypes and more of the non-recombinant genotypes are coming out of it. So far exceeding 50% there. So following that, we went through some of the math and talking about recombination frequency and recombination fractions. So recombination frequency is basically the number of recombinants divided by the number of progeny times 100. And we talked about what this actually represents is not just how much recombination is going on, but often we use it as a measure of physical distance as well too, to look at how far in terms of base pair we are in between genes. Is that a one-to-one kind of correlation? No. But does that ratio frequently mean something? Yes, it does. The more recombination that you do have, the further apart those two genes normally are on a given chromosome. Okay. So a couple of terms that I did not mention last time about orientation of these two genes on the chromosome. And orientation specifically, which alleles are paired together on the chromosome. These configurations of linked genes, we can have either coupling or repulsion. And coupling is more frequently called cis configurations, often when you kind of talk about. And here, this is where dominant alleles are found on one chromosome and mutant alleles are found on the other chromosome. So your book frequently has this terminology that's wild type alleles. And it's something that most evolutionary biologists hate to use. Because what is the wild type allele? It's really the most common allele in the population. And then we call anything that's not that dominant, that wild type allele, the mutant allele. Not necessarily the best terminology here, because in this, we really are talking about the dominant allele, because it is the one with the plus signs. So in this coupling configuration, we have those two genes, the dominant alleles paired together on one of the chromosomes, and we have the recessive alleles paired together on the other chromosome. This is the situation that we've been talking about so far entirely. Every one of the graphs I've showed you has been in this coupling kind of formation. And that you get the two dominant phenotypes coming out of the progeny. And if you're doing your test cross progeny, it would be that purple thorax, black precum, the recessive traits, the two recessive traits coming out of that. However, we can also have this trans configuration, this repulsion. And that's where the dominant, one dominant allele and one mutant allele of different genes are found on the same chromosome. For instance here, you can see that the dominant allele at the P gene is found with the recessive allele for the B locus there. And we're crossing it with something else here entirely that has the dominant allele of the B locus and the recessive allele of the P locus together. Now, what's the importance of this? It really depends on if we're talking about discrete or quantitative phenotypes in general. If we're talking about quantitative phenotypes, and we have a dominant allele and recessive allele that do different things on the direction of the phenotype, say we have one particular gene, so two genes that affect plant height, and one of the alleles increases plant height and one of the alleles decreases plant height, you can see how the configuration of those two alleles might matter a lot. If the two alleles that increase plant height were together, you get a nice additive effect. However, if these two genes are close together and you have alternative effects at those two genes, you might get no increase in plant height whatsoever, despite the fact that you have two genes that affect that quantitative phenotype in that, on that same chromosome. It also looks a little bit different when we go through these test crosses that we were talking about last week. So this is the situation we've been talking about so far with that cis or coupling configuration right here, where those two dominant genes are on the same chromosome. So remember that when we go through gamete formation, we don't get any recombination necessarily, or very limited amounts of recombination between those two genes because they are fairly close together. So the non-recombinant gametes are going to be the same gametes that you have in that parental generation, with the two dominant alleles on one chromosome and the two recessive alleles on a second chromosome. And then on a far lesser frequency than those two, we're going to get these recombinant gametes whenever we have some sort of recombination event occurring between those two genes. If we're doing a test cross over here, then we have a double recessive homozygote that we're crossing with. So there's only one possible gamete we can get out of that, and that's those two recessive alleles on a single chromosome. And this is the situation we've basically been talking about so far that we went through on Friday, where we have more non-recombinant progeny down here, the same ones as the parental generation, than we do the ones that have those recombinant phenotypes. Now, how does this differ if we have a trans configuration up here? We're doing the same test cross, so this individual over here has not changed at all. That's that double recessive homozygote over there. But if you look here, whatever the common gametes are going to change in this scenario. You still get your parental gametes. If there's no recombination, those are the ones you're going to get out of this. So the dominant allele at the P gene and the recessive allele at the B gene, or the recessive allele at the P gene and the dominant allele at the B gene. Those are your two non-recombinant gametes. So if we're putting that through, our frequencies in this F2 generation are going to change dramatically. Again, we get the parent phenotypes coming out of this cross more often because these are the non-recombinant progeny. And we get these other recombinant phenotypes coming out less often out of that particular cross. So the 4 phenotypes that you actually get out of this cross, they don't change whatsoever. It's just the ratio of those particular phenotypes that you're getting for those different linked genes.
So is there like a constant that you would like plug in to figure out how many are likely to be recombinant progeny? Or?
So almost always we'll either, 2 different ways we can do that. Like 1, I'll give you the recombination rate that's actually going on. 2, I'll make you calculate the recombination rate that's actually going on from the frequency of progeny in the F2 generation. And I'll have to tell you that's either a cis or trans configuration. This is more something for when we're looking at bigger data sets later on that we have to take into account. And reasons why, if we're doing kind of these crosses in the QTL mapping format I'm going to talk about on Wednesday, we might not find some of the genes coming out of that is because they're in these weird allelic configurations that mask the effect of the phenotypes. Okay. So I think one important thing that I often get people tripped up on is the difference between what happens when you have linked genes versus epistasis. And one of the reasons this can be confusing to people is because we talk about often deviations from the 9:3 to 3:1 ratio in the dihybrid cross, both for epistatic interactions and for these linked genes. So if you remember what epistatic interactions actually were, they were when multiple genes affect the same phenotype and often bring out these novel combinations of phenotypes. So the examples that I gave 2 or 3 lectures ago where the sweet peppers, where you had the red, the cream, the yellow, and the green sweet peppers. That was an interaction at 2 loci. We talked about the dogs and the yellow versus brown and black pigmentation. And these are all different examples. And of course, we talked about cynogenesis for an entire lecture. And we talked about all these different kinds of epistasis that actually occurs here, and how the kind of epistasis that we're looking at really depends on your deviation, that ratio, in those genotypes in the F2 generation in a dihybrid cross. So how can we tell the difference between linkage and epistasis? Well, first of all, epistasis is often only acting on one phenotype necessarily. For instance, in cynogenesis, it's the ability to produce hydrogen cyanide. In the dog pigmentation coat color, that's a dog pigmentation coat color. In the pepper example, it's the endosperm color of the pepper. But sometimes it's hard to tell apart when you have one phenotype versus multiple different phenotypes here. And we can think about this in terms of morphs. So in a dihybrid cross with no linkage and 4 morphs and the genes on different chromosomes, we get out that perfect 9:3 to 3:1 ratio in F2 generation. However, when we have these epistatic interactions, we get either 3 or 4 morphs coming out of this, typically in one phenotype, if you can tell what that phenotype is, and they deviate from the 9:3 to 3:1. And they deviate in the way that's dependent on what type of epistasis is actually coming. My one being, for the cynogenesis one, being the 9:7, where you get most of the individuals that have at least one copy of the AC and one copy of the dominant LI gene that create cynogenic. And then you have all these other genotypes that add together that are not cynogenic in that case. Okay. So if we do a dihybrid cross with linkage with those 2 genes on a single chromosome, and there's no recombination whatsoever, we only get 2 different morphs coming out of that. And that's the one corresponding to if you have a single dominant allele at each of those 2 genes, or you have a double homozygous recessive individual. If we add recombination into that, though, we're going to see that most of our progeny are these non-recombinant progeny that we have. But every so often, we're going to see some recombination level in them, depending on how far apart those genes actually are. And we're going to see a limited level of these 2 different phenotypes right here. So this corresponds to one of the epistatic configurations that you have in here. Now this normally will produce 4 different morphs, so not 3 different morphs. If you look up here, if you're combining those 2 things together, like a 9 to 6 to 1 in epistasis, you'd only see 3 morphs in that. However, if you had this down here, you would see 4 morphs with 2 of them in lesser configurations, or lesser frequencies. The important thing when going through this is to create a model for yourself. Write this out, what the gametes are actually doing here, and what you think their individual phenotypic effects actually are.
So with the dihybrid cross with linkage, no recombination, if you were to do that, I don't know, and get 16 progeny, what would your ratio be?
In a test cross or a dihybrid cross?
In a test.
In a test cross, it'd be 50-50. In a dihybrid cross, I think it is 75% to 25%, if I remember right. But I can draw it out if you guys would like me to do that. How many people would like me to draw out?
Yeah, and what kind of epistasis would that look like?
What kind of epistasis?
Like when you have a dihybrid with the 75-25, what kind of epistasis does that look like?
So
let me go back to
that would not look like any epistasis right there.
Okay.
That would look completely independent of epistasis, which is kind of the point of it.
I think I'm caught up on like what tends to look like epistasis when it's not.
What tends to look like epistasis when it's not?
And
Simply, it doesn't, like none of these combinations will look precisely like epistasis. The thing is, it's a deviation from that 9 to 3 to 3 to 1 ratio. And that's what I say I harp over constantly at a time. So that's why I want to make sure you understand that you can have deviations from that 9 to 3 to 3 to 1 ratio from several different things, including both linkage and epistasis. Okay. So we have A, B, A, B. Okay. So this right here is a test cross. Dihybrid or test cross? Let's do a dihybrid cross right now. So if we're looking at the configuration of these, this with no recombination whatsoever. These. So with no recombination whatsoever, what are we getting out of this? We're going to get the parental gametes back again. So we're going to get A, B as a gamete, and little a, little b as a gamete. Out of this, what are we going to get out of meiosis? We're going to get, there's only one option that you possibly can get here, little a, little b. So let's do this in that test cross that we were talking about right there, or in a Punnett square. Dad, Mom, I'm on Jackson. Okay, good. Dad on the top. So we have 4 different typically we think about 4 different allelic combinations that come out of this. However, in this case, we're only going to get 2 of them: big A, big B, little b. Does everyone understand why those are the only combinations of gametes I can particularly have there? Good. In every single one of these gametic combinations, when we get this out of there and pull out our 4 different gametes that we can have, it's going to be little a, little b. Now you can imagine that all 4 of those phenotypes are going to be exactly the same. Right? They're always going to be what's coming out there. These 4 phenotypes are all going to be the same as well too in this test cross. They're all going to be that double homozygote recessive individual right there. All right. Here, they're going to be exactly the same as in that first case again. You get 1 dominant allele, 1 recessive allele, 1 dominant allele, 1 recessive allele, both A and E locus. All the way down. And again, same thing here, AA, BB, all the way down. Great, right? So basically, in this test cross ratio, we're at 50% and 50% there. Okay. What happened?
With the top 50% not, you drew the lines, which means it could be varying, right?
Yes.
So
You're completely right in this scenario. It is not varying whatsoever. That would be it here. In a dihybrid cross, when I'm doing this, it would be the lines. Because you would still get one down the allele out of it, but you get underscore.
What's the difference between a dihybrid cross and a test?
In a dihybrid cross, we're crossing two double heterozygotes together. In a test cross, we're crossing a double heterozygote with a double recessive homozygote. So test cross is right here. Dihybrid cross. Sorry about my handwriting, y'all. We're going to self it like that, or we're going to cross it to a different individual if it's something you can't self.
Those ratios are non-recombinant.
Those are all non-recombinant. So if we started getting recombination out of this, we wouldn't be getting equal frequencies anymore, and we couldn't use this nice little handy-dandy thing right there. But if you picture coming out of this multiple times, what we normally would have in this, right, is instead of having those phenotypes on there, we would have we'd have something like that, right?
Okay.
So
that results from like crossing over.
That results from either crossing over, or if you have two genes on different chromosomes, that results from segregation at meiosis. So that's why at meiosis, because we have Mendel's laws of segregation, we get perfect segregation into those four gametes, and we can use these nice Punnett squares. If they're on the same chromosome, all hell breaks loose, because we don't get recombination happening that often. And if you're going to do this in terms of proportions, to make it look something like that, if you're having the size proportions for each one of these. A little bit more understanding?
So with a regular, just a normal cross like we did at the very beginning of class, that, let's say you could have one gene on like the 15th chromosome and another one on like 21st, and they both affect each other, but they assort independently. So that's why you get the ratios that you have.
So they don't even affect each other in the original Mendel's crosses that we were doing. If we were doing something for P color and for P shape, like round versus wrinkled, those phenotypes don't affect each other whatsoever.
I mean, they feed into the same biochemical pathway. You get this, like,
yeah, for the epistasis things, those two genes would affect each other, and that's the difference right there for why we deviate from that 9 to 3 to 3 to 1 ratio. Okay, so what kind of question am I going to actually ask you on a quiz or a test for this kind of question? Or what is anybody else going to ask me for that matter? As I said, I could give you a recombination frequency. So geneticists have determined that recombination frequencies between two genes in cucumbers is 16%. How can you use this to predict the information and the results of this particular cross for these two genes that are on the same particular chromosome? So if you think about this cross that we're doing a test cross again. We have that double heterozygote that we just talked about, exact same thing as before, double homozygote recessive individual. We got those two non-recombinant gametes that we're going to get more of here, but that recombination frequency there is telling you something. We know that recombination frequency is the number of recombinant progeny divided by the total number of progeny. So if you're getting 16% of your progeny that are recombining, what you should get is 16 out of 100 individuals that are these recombinant genotypes. Now, we can't say anything about which two of these recombinant gametes there are. And really, in practice, they should be equal. Because if you have a crossing over event in a particular location, one of your gametes is going to be the big T, little d. And one of your gametes will be the little t, big D, simply because there's a crossing over event between those two. And there has to be that 50-50 ratio there. So if we got out of 100, or if we're just going for frequencies, we know that 8% has to be each one of these recombinant gametes right there. And we can bring this straight through into that test cross that we have up here that I showed you before. And through this predicted frequency of recombination, we can use our product rule that we had before. And we can say that for this particular individual, the gametes that we're going to get 42% of the time we're going to get that big T, big D gamete up on top here. That's because that's our gamete frequency from our male or female parent, doesn't matter. From our other parent, 100% of the time, we're going to get that little t d allele. What's the frequency that we get both of those two gametes? Multiply them together. 0.42 times 1, 42%. So it's basically you're going to get this frequency of progeny out of it. 42%, 42%, 8%, and 8% of each one of these recombinant gametes. Now what would I do if I saw this on the test? I would pretty much write out this entire thing right here for the different frequencies that you have for each one of the gametes and go through it that way and look at it in that kind of statistical framework there. Okay. Hunter.
Is there any way we could like work out something like that in class before the test?
We could do it in office hours
okay.
at any point if you want to work that out. I also recommend just going back to this example in the book, and there's like 5 more examples of the exact same thing in the book. It's fairly a frequent thing. Every time I give you that recombination frequency, divide it by 2, and that's your 2 recombinant gametes right there.
How would you do it backwards?
How would I do it backwards?
How do you, like, you give me that and say find the recombination frequency?
Exactly. Do you remember last time when I did the pea flower example? So it was the yellow and red pea flowers with the long and short pollen.
I think I have that right. Yeah.
That's exactly what I'd give you right there. I'd give you the number of different progeny that you have. So I'd tell you that you have this deviation in an F2 in a test cross, and I'd ask you what the recombination frequency would be of that. So that's the example I gave you in the last lecture. Okay. So, yes?
Is there like a practice question that we could do in the book? Are there practice questions in the book that focus on recombination?
There is about 40 practice questions for each one of these chapters in there. So there will be a bunch of these there. And I will send you guys a link to 5 or 6 that I think from the chapter you should be able to know. But basically, that last example that I gave you right there, word for word, that's something I'll probably give on a quiz or test. Something like that. Okay. So what we've been talking about before, we have these chi-square goodness of fits tests that tell us if our expected ratios match the ratios we would expect from a given cross, or the probability that that would actually occur. But we have this problem in nature that most of the time when we go out to a population, we have no idea what the frequency or what the pedigree looks like in that population. We have no idea who the moms and dads of these particular individuals were. So we can't create those expected ratios for a chi-squared goodness of fit test. We have no idea who's the male and female, if it should be a 3-to-1 ratio in these particular progeny or whatnot. But what we can know in that particular population is if 2 different genes have alleles that are segregate together more frequently than we'd expect by chance. This is a different chi-squared test. This is the chi-squared test of independence. And all they give it to you with a cross here, so you could do a chi-squared goodness of fit test. We're going to do this like a chi-squared test of independence. I'm going to show you how that differs to create these expected ratios. Why are these both called chi-squared tests? Because you're going to use this equation for both of them. That observed minus expected squared over expected for each one of the different phenotypic categories. Okay. So in this particular situation, let's forget about this cross altogether right here, so we have no expectations of what's coming out here. We go out in nature, and we're looking for cockroaches. So we go back into the shed behind my house, and what we find is 63 cockroaches with a brown body and straight wings, 28 with a brown body and curved wings, 33 with yellow body and straight wings, and 77 with the yellow body and curved wings. And we say, hey, we know these are Mendelian genes right here. I wonder if these 2 genes are on the same chromosome, or are these 2 genes on separate chromosomes? How are we going to look for this? We want to see, just like in our epistasis example before, if these ratios are off in here, if they're what we'd expect out of a particular segregation of alleles. And we can create these particular expectations ourselves by looking at this, by adding up the rows and columns and having the total number of progeny right here. So if we create this particular chi-squared box that you have, and you add the different columns, so 63 plus 28 here, and that 33 plus 77, and then across as well in the same way, we can create a given expectation. So from these progeny ratios that I expect right here, the number of progeny that I have, and the total number looking at each one of these genes independently, we can try to figure out what our expected ratio is of when we get one big Y allele and one big CV allele here. So what would that be? Let's look at this in a gene by gene basis. So if we think about how we get this particular one, we can look at, you add up all these individuals here, and that's all the chances that you get that big Y allele from your dad. How often do you get a big Y allele from your dad then? You get that 91 out of 201 times. Okay, so to get this particular genotype, we have to get a big Y allele from our dad and a big CV allele from mom. So what's the frequency we get that CV allele? Well, it's 63 plus 33, we have that big CV allele, over 201. So what's the frequency that we get both of those two alleles there in that cross? Well, it'd be 91 over 201 times 96 over 201 to get the frequency of those two things together in the same individual. And from that, and then multiply it out times the total number of progeny, because this is going to give you a ratio right here, we'd predict that from these ratios of our individual genes in there, that we should get 43 and a half offspring for this particular square right there. And we can do the same thing where we establish predictions for each one of these boxes based on the inheritance patterns of those individual alleles at different genes, and then compare that to what we actually see for an observed ratio. So same thing, I'll go through one more for you here. So what's the probability that we get one big CV allele and one little Y allele out of this? So that would be this box right here. So again, what's the probability that we get a CV allele? 96 divided by 201 altogether. What's the probability that we get a little Y allele? 33 plus 77, so 110 over 201. It's probability we get those two things together. Let's multiply them together, get a particular frequency there, and multiply that times the number of progeny that we actually have. We expect to get 52.5 progeny in that particular cross. You also have a couple examples of these in the back of your book, and you should probably run through these as well too. So now this part is a part that you guys have done already. We have observed ratios, or observed numbers up there. We have expected numbers. We can simply put them together with that formula over there. So top box, let's go through one of these. We observed 63 individuals. We expected 43.5 from those calculations of allele frequencies at individual genes. We can put these all together and add these together over here to get a chi-squared. That chi-squared is 30.7. What does that mean? Where does that put us in a probability ratio? So remember, we have to go to our probability level table over here to understand how absurd it is that we would get a chi-squared, that it was that high, what the chance or the probability is given our expected ratios that we have at those two particular genes, that these are linked. In this case, our degrees of freedom in a chi-square test from independence, that's the other thing that differs slightly from that chi-square goodness of fit. So because we're looking at two genes independently, and those genes can only vary in two ways each, basically if you don't have one possibility, you have to have the other possibility. You have to take away a degree of freedom for the rows and the columns there. So basically if we have these same two genes, your degrees of freedom for this entire thing is going to equal 1. For the goodness of fit test, if we're testing at 9 to 3 to 3 to 1 ratio, and we have 4 different phenotypes there, and we have those expected ratios from our Mendelian crosses, then we'd still have 3 degrees of freedom. So where does this fall? We have 30.7 for our chi-squared, and I said we have 1 degree of freedom. Followed over in the table. At 0.001% of getting that particular observed values, expected by chance, we get a chi-squared of 10.83. Ours is 30.70. So what this is saying is this is very unlikely to happen just from the ratios that we have at these independent gametes if they were segregating independently. So they are not segregating independently in this particular case. Quickly, Ollie.
I'm sorry. Could you run over again how you got? So did you just add each row like that and then down?
I just added across,
Okay.
and then I divided these particular numbers by the total number of progeny to get the expected ratio for just individual genes. Just looking at the expected frequencies of the alleles. But I have to get you guys started on a quiz now because there's a test coming in after you, and we have to be done by 9:50 today. Well, this is a shorter quiz.
Okay, so you said it's